Hi, everyone. Welcome to Unit 2 of the course. Good job on finishing the first unit of the course. As I said, the course is broken down into three parts, and the materials are not cumulative. So we kind of start brand new in Unit 2. And we have 4 lectures in Unit 2, starting with the part 1 of nervous system, which mostly deals with the cellular level of the nervous system. Okay, we're going to learn about the different parts of a neuron. We're going to learn about how neurons communicate with each other, as well as how signals are regulated. Um, when you try to go from one neuron to another. So that is the focus of nervous system part 1. And then there's nervous system part 2, which is more like the gross anatomy. So like the different parts of the brain, of the spinal cord, and so on and so forth. And then lecture 7 will be on sensory organs, which talks about like the eyes, the ears, uh, how they work, as well as taste, basically our 5 senses, and the physiology dealing with these senses. And we'll wrap up the unit with digestive system, okay, which is kind of straightforward out of all of these topics. Okay, so as with the first unit, you will have one quiz in this unit, which is covering material from lecture 5, and then you will have a lab, 2.5%, and then there's going to be the test, which is 6, 7, and 8. Lecture 6, 7, and 8. That will be 20% as well. Okay. This is a difficult lecture. There's a lot of moving parts, a lot of new abstract concepts, and we will be taking a lot of notes. So have some papers with you, and again, use colors because we have many different things we need to highlight, and it makes it easier if you have colors. And also in this unit, there will be a lot of diagrams that we have to label and color together. So please try to print the study guide. That's where the diagrams are. Even if you don't have a printer, right? Maybe try to do it at a library or something, because you really, really need to have those sheets. It's very hard to learn the parts if you don't have a physical sheet to work with, as I teach you the different parts and stuff like that. And get some crayons, get some markers. So yeah, without further ado, let's start with the nervous system part 1 right here. This picture is it's the end of a neuron, and you can see these blue stuff that are kind of coming out of the neuron. And you will have a better understanding of this towards the end of the lecture. But in case you're wondering, that this is what the ends of a neuron look like. So last semester we learned about the different types of tissues. There is muscle tissue, there's connective tissue, epithelial tissue, right? And the last type of tissue is the nervous tissue. The nervous tissue contains neurons. So neurons are basically like a nerve cell. Nerve cell. These are responsible for creating and transmitting electrical signal. Okay. Now you will see electrical signals are called impulses. Yeah. You might see that coming up again and again throughout the lecture. So these impulses, these electrical signals, is what allows neurons to communicate with each other and say for the brain to send a signal to the hand so that the hand can move. Right? So all these communications are done through electrical signals that are being passed along the neuron. So neurons are very precious, as you probably know. They don't divide, they don't undergo mitosis. If you lose a neuron, they don't come back. Okay. And so we want to protect them, we want to support them as much as we could, which is what the glial cells are for. Okay? Glial cells. Glial cells are also known as neuroglia. These are cells that would support, protect, and kind of help nourish the neurons. They outnumber the neurons 9-1, and it takes up more than half of the volume of the brain. Okay, so there's a lot of these glial cells, certainly much, much more than the neurons themselves. And we will learn the different types of glial cells along the way. This picture just shows you some of them. There's something called like ependymal cells, oligodendrocytes, so on and so forth. Some of them you will find in the central nervous system, which includes the brain and the spinal cord. We will talk more about these later, okay? Like, I know it's a lot of information on the slide, but this is just an overview. We're gonna go into, much, much more detail as we progress. And so brain is up here, spinal cord goes down, right, all the way to your lower back. So that makes up the central nervous system. And everything that's outside of the central nervous system is going to be your peripheral nervous system. Right? So like, you would have a nerve that goes down your arm, right, to your fingers. So when you touch things with your fingers, you feel the roughness of the paper or something like that, or like if it's smooth, right? The feeling, that texture, that sensation goes up the arm and goes to your brain, right? That travels along the peripheral nervous system. So brain and spinal cord, central nervous system. All these things are extending out from it. That will be part of the peripheral nervous system. So again, we'll talk more about that later on. So the neurons themselves, like I said, they don't divide, which is a little bit puzzling for people sometimes. So like if the neurons don't divide, then how can people get like brain cancer? This cancer, after all, is uncontrolled cell division, right? So it turns out the cancers came from the glial cells. Okay, so glial cells, unlike the neurons, they are capable of doing mitosis, and when they uncontrollably divide, then you get different forms of brain cancer, depending on which of these glial cells are dividing rapidly. So, for example, if the astrocytes are the ones that are dividing in an uncontrolled manner, then the person can get something called astrocytoma. If it's the ependymal cells that are becoming cancerous, then the person can get ependymomas. So there are different kinds of brain cancer depending on which type of neuroglial cells is doing the division, basically. Okay. Let's go ahead and talk about the structure of a neuron. Now, in your study guide, there is a picture there. So I want you to find this one. Now, I'm just actually going to copy it to the class note, so I have it all on the same page. And then I can export it at the end of class for you to see it, right? So there is the neuron. I'm just going to put it here. There we go. So this is a structure of a neuron. Okay. Do all neurons look like this? Nope. This is just like a typical one, right? You might, I mean, in this picture, you see like a long tail here, right? But sometimes neurons, like they have like two tails like this. So we're not going to go into too much detail of the different kinds. We're just going to focus on like a typical neuron. All right. So here, this is the cell body. Cell body. Nothing unusual about it. This is where you will find contains um, typical organelles. Right? Like you will find your mitochondria, you will find your Golgi body, and so on and so forth, right? And the neuron typically contain more mitochondria than other cells because it requires so much energy to do the information processing, to transmit the information, and so on and so forth. And of course, being a eukaryotic cell, you're going to have the nucleus in the center. All right. Nothing unusual so far. Coming off from the body, you're going to have these kind of like tree branches. Some people think they kind of look more like, ice crystals, right? Branching off from like a snowflake or something. These are called dendrites. Dendrites. Okay, so these are extension from the cell body, and their function is to receive signals from another cell. Why is there this thing there? That's so weird in a way. Okay. Receive signals from another cell. Okay. Usually a neuron, but doesn't have to be. And I'll explain what that means later on. Okay. So it will receive signal from another cell. This other cell that's passing on the signal is usually another neuron. But like I said, it doesn't have to be. It could be something else. And I'll tell you what that something else is later on. Okay. It is not uncommon for, why does it not let me change color? Okay, I'm gonna have to restart the program. Something's weird going on here. Give me a second. All right, let's see if it's better. So, when the signal arrives, they could be excitatory signals. These green arrows, we call them excitatory. Signals. Okay. Think of them as like an on switch. Okay, it's telling the neuron to go ahead and pass this signal on. Okay, turn something on. Okay, move the hand. That's an example of like an excitatory signal. And at the same time that it is receiving excitatory signal, it could also be receiving inhibitory signal. Which is to tell the neuron to, stop something, to not do something. Sometimes we have to inhibit our body to do certain things, right? Very simple example. To move the arm here, you have to stimulate the bicep, right? But then you have to like relax the tricep, right? So like you have to inhibit the movement of the tricep in order for the bicep to move. So, you know, excitatory signal and inhibitory signal. Okay. Another example, like sometimes you have to suppress yourself from saying something, okay, or from behaving in a certain way because, you know, the situation you are in is not appropriate to do certain behavior. Like if you are in class, right, like in an actual physical class, even if you are very bored, you know it's not proper to stand up and say, oh, this is so boring, right? Like you can't do that, right? Even though you want to, but you can suppress yourself to not do that because it's not the right place to do that, right? So, you know, you have these inhibitory signal stopping you from, you know, certain impulses in situations where it's not appropriate, right? So it's not always passing on a go signal. All right, sometimes you have to suppress certain things as well. So at any given time, the neurons are receiving these excitatory signals and inhibitory signals, and in the cell body, right, this is where you are going to interpret those signals. You're going to count them up, right? You will add up all the excitatory signals, you will add up all the inhibitory signals, and you see which one is stronger. Okay? And then you're going to proceed with that. Do you understand? Right? So it's just a number game. Right? Do I pass the signal on? Do I not pass it on? All right. Let's say we do pass it on. So the signal is going to go down this very long tail that is right here. Okay? So this tail, super long one, that I'm coloring right now. I'm only coloring the inside, okay? And this one here, that is called the axon. And it's right here, this arrow here, axon. So it carries the signal, the impulse, towards the axon terminal, which is right here. The axon terminals are like at the end of a neuron. Axon terminal. Which sends signal to another cell. And this other cell is, again, usually a neuron, but it doesn't have to be. And I'll explain again later. Okay? It will make much more sense when I give you the full picture.
I have a question.
Yes, please.
All cells make a long line to the brain, and
All cells make a
make a
long,
long line. Yeah,
to the brain?
yeah. But the
No,
axon pass through all the way to the brain.
not necessarily. Okay. Hold that thought. We'll come back to it and I'll show you how exactly it's like, you know, connected. But no, short answer is no. Like this axon doesn't necessarily go all the way to the brain by itself. Okay.
Okay, thank you.
Yeah. So surrounding the axon are these you know, kind of like you know, like a hot dog, I guess, or Twinkies. Twinkies probably better analogy. Okay, so like you have these. If you draw it in 3D view, you have these waxy membrane that goes around the actual axon. So the axon is right in the middle. Okay? And then you have this waxy coating that goes around the axon. Okay? So that is called the myelin sheath. Myelin sheath. Okay? The myelin sheath is created by the glial cells. Created by glial cells. And depending on whether you are in the central nervous system or if you're in the peripheral nervous system, the kind of cells that are involved would be different. And I'll tell you about them later on. Okay? But you know, you will surround them, and the purpose of the myelin sheath is to speed up conduction. Okay? Like the insulation that we put around copper wire, it allows the signal to not be lost along the way, and it allows it to go faster. But we do not wrap the entire axon. We have to have some gap here. So there is a gap there. There is another gap here. Right? There's another gap. These gaps, they are called nodes of Ranvier. Okay? Nodes of Ranvier. V-I-E-R, sorry. Okay. This is where ion exchange occurs to propagate the signal. Okay. We will talk, I keep on saying this, but we will come back to it with another note that tells you what this actually means, like what kind of ion are being exchanged and how that helps propagate a signal. Propagate just means like to pass on, right? So if you think about a signal like this, the signal will jump from node to node to node, okay? And then it gets passed down. So this jumping action allows the transmission to go much faster than it otherwise would. Okay.
A quick question.
Yep.
Does the axon go right to the end of the axon terminal?
Yeah, it goes all the way. It goes all the way.
Okay.
Yeah, right here. So you can think of it like this. Right? You have one neuron here, and then you're gonna have another one here, and then you have another one, right? So like the signal is just gonna go from one to another, and then it's gonna jump like that. And over here, this is basically an axon terminal passing the signal to what? Here, I'll call them 1, 2, 3, okay? This would be axon terminal of number 1 passing the signal to, what should I write here?
Number 2, right?
Yes, what of number 2 though? Which part of number 2?
The nodes of Ranvier.
Okay, good
Cell
try.
body.
What's that again, Mohammed?
Cell body.
Okay, also good try. Let's take a look here. Which part receives the signal, guys?
The
Dendrites.
dendrites?
Yes. Okay, so this is gonna go to the dendrites of 2, right? Do you understand? Now you said cell body, right, Mohammed? Sometimes that can happen. It's possible to connect directly to the cell body as well. But then, you know, most of the time it's axon to dendrites. Okay. And again, I keep on saying this. As we'll see later on in the lecture, that connection between the axon terminal and the dendrite, we have a name for it. It's called a synapse. You probably have heard of this before, right? Like a synaptic connection. You have, you know, billions of connections in the brain. All these synaptic connections. It doesn't have to be a one-to-one connection either. One neuron can, you know, be connected to multiple other neurons. Okay, so that is an overview of a neuron, of a typical nerve cell. Any questions before we proceed?
I have a good question.
Yeah.
What force lead axon go node to node and it going through to axon terminal?
Okay, so how come it jumps from one node to another node, right? Yeah.
Yeah.
We're gonna have a super long node coming up for that. Probably after the break.
Okay.
Yeah. Anything else? You guys with me? Okay, fantastic. Moving on. Now, it turns out the same thing is going to be called different names depending on whether you find it in the CNS, central nervous system, or the PNS. So what's CNS again? CNS is the central nervous system. It only contains two things, right? The brain and the spinal cord. If you find this structure in the CNS, we give it one name. But if we find the same thing in the PNS, then we give it a different name. It's like, you know, sometimes in Canada and in the US, we call the same thing different things, right? Like, you know, when we finish a meal at a restaurant, we ask for the bill, right? But then they say, give me the check, right? So same meaning, but different things, right? So same thing, but different names, I mean. So here, if you have a bundle of axon, and you remember axon, right? We just label it, it's like that tail, right? But this is like super small, right? And microscopic, right? But if you have a bunch of them, okay, so look, each of these is like an axon. If you have a bunch of this whole bundle, in the PNS, we call that a nerve. Okay, a nerve. Okay. You hear people say you struck a nerve, right? That's the nerve that carries the signal to the brain and the spinal cord, or it carries signals away. Okay, so a nerve basically carries signal to, oh my goodness, to and from CNS. Okay. You want to move your finger to write the notes? You got to send the signal through the nerve to your fingers, to your hand muscle. Okay, you touch something sharp and it, you know, you feel the pain. That pain is because the signal goes through the nerve and then it carries it to your brain and the spinal cord.
Question.
Yeah.
So it starts in the central nervous system and then it like goes off into the peripheral nervous systems, like you said, like your finger, your toe, or whatever.
Okay, interesting question. Not always, that's the short answer. In 3 slides, we're gonna take a note and it will explain that. Okay, 3 slides. Just hold on. Now, once it's inside the brain and the spinal cord, the same thing, the same bundle of axon, okay? We give it a different name. We call it a track now. Okay, track. So a nerve and a tract essentially is the same thing. They are a bundle of axons, but the location in which you find them gives them different names. Okay. These axons are covered with myelin. Myelin sheath, same thing, doesn't really matter. They are for insulation, and the job is to speed up the conduction. Okay. Speed up the conduction. So here you can see there is a single axon there, single axon. And then you bundle them together, and then you bundle them, more of them together. That's basically what it is, okay? It's not gonna be a single nerve, it's too delicate. Right? Like if it breaks, then you completely sever the connection, right? So you need to have a bunch of them. You gotta surround them, you gotta wrap them up and protect them.
I have a question.
Yeah.
So we're talking about the peripheral nervous system in the first bullet point. So I'm a bit confused why it carries the signal to and from the central nervous system. Or is that like 3 slides?
Yeah, third slide. Just hang on there. It will
Okay.
make sense. I promise you. All right. Why don't you guys? Oops, sorry. One more slide. So the myelin that I was talking about, the yellow thing that wraps around the axon, that speeds up conduction, and it's created by glial cells. Depending on which part of a nervous system we're talking about, then the glial cells are going to be different. So in the PNS, in the peripheral nervous system, the name of the cell is called Schwann cell. In the CNS, it's called the oligodendrocyte. Okay, they're just different names. Just write this down on a sheet, okay? Like for the quiz. I know it's hard to memorize all these names, okay? The point is just to know that there are different cells that plays different role. We talked about multiple sclerosis last unit. Remember the autoimmune disease? In multiple sclerosis, these cells are being attacked by your immune system, the Schwann cells, and you lose the myelin. Once you lose the myelin, you cannot communicate with the body anymore, right? Because the signal is not being transmitted, and that's why it causes paralysis. Okay. So, please try this next question. Okay, let me launch the poll. It's asking from one neuron to another, right? It's not asking for like, down the neuron, right? I think maybe that's why people are getting confused. So over here, look. Right. To go from one neuron to another neuron, the signal is going to have to pass through the synapse. Okay? The synapse. A lot of people chose the axon, okay? But axon is like, going down the neuron, right? It's not, the axon's not going to cause the signal to go from one neuron to another. The axon is transmission within the neuron itself, right? Maybe I wasn't being very clear earlier, but that's what this means, okay? Synapse. Again, this is one neuron. This is another neuron. To go from one neuron to another neuron, it got to go through the synapse. Synapse. Okay, this one, purely factual. You probably will see something like this on the quiz. What is it? Okay. Yeah, just look it up, right? Okay, that's good. That's good. See, don't you wish every question is like that on the test, right? It would have been much easier, right? So that's the answer. Oligodendrocyte. Oligo means like little star-shaped cell. That's literally what the name means. Oligodendrocyte, little star-shaped cells. Okay, the third slide that we have all been waiting for. So please take out your study guide again if you have it. It's this box that I'm gonna fill in, okay? Draw a diagram that shows the flow of information in the nervous system. I'm gonna draw it out. Now you can put it in here now, or you can do it on a separate piece of paper and then reorganize it after class. Whatever you want, doesn't matter. The picture is going to, so like if this is your paper, we're gonna start over here, we're gonna go over here, and then we're gonna go over here. All right, so like plan your space accordingly. All right, so this is flow of information. In the nervous system. Right. So I'm gonna go through this using a reflex as an example. Okay, reflex are things that your body automatically knows how to do without prior learning or conscious thought. An example of a reflex that we all probably had an experience of is, let's say there's a candle here, okay? There's a candle and you accidentally touch it without realizing it, okay? It's probably more common with like a hot surface. You don't know something is hot, you touch it, and then your hand like, retract, right? In fact, you retract your hand even before you realize you got burned. Right? But let's just say, you were not careful, you weren't looking, and you accidentally put your hand on a hot candle. Okay, so between the time you touch the hot surface and the time your hand retract, like what's going on, right? Like how are the signal going to the brain and so on and so forth, right? So I'm gonna go through that with you. So to begin all this, you need to have some kind of detector, right? That's capable of detecting the change in temperature. So we call that a sensory receptor. And you just have to trust me, they kind of look like this. Okay? This is a sensory receptor, which is responsible for detecting some kind of stimulus, some kind of trigger in the surrounding. And there are different kinds of sensory receptor in the body. That's lecture 7. But for now, right, like in this case, we would have to have a receptor that's sensitive to temperature change. What other things can you feel with your hands besides temperature?
Texture.
Texture, that's right. So that's another kind of receptor that's needed. What else besides texture and temperature Give me two more, guys. What can you feel with your hand?
Sharp or dull?
Yeah, that's also kind of texture-ish.
Weight?
Yeah, pressure, right? Like if something is pressing on, doesn't have to be the hand, but like, like you can tell the difference between someone poking you with a blunt end or the sharp end of a pen, right? That's basically pressure. So there are pressure sensors. Another one, last one.
Soft and rough.
Yeah, that's also texture. Yeah. Come on, this one is really obvious, guys. If I say it, you'll be like, oh yeah, of course. It's probably too obvious that you didn't even think about it. Again, not just your hand, right? But like throughout your body, the skin. Right? What can you feel? It's not a pleasant feeling.
Like if it's wet?
Okay, yeah, interesting. That's not what I
Pain.
have in mind, but could be. What's that? Pain.
Pain.
Okay, thank you. Yeah, pain. Okay. So you have pain receptors. You have thermal receptor, temperature, right? Thermal receptor. You have mechanic receptor that detects change in pressure and stuff like that. Okay. So there are different kinds of receptor. You don't have to write any of them down. You will learn them in lecture 7. Okay. But for now, the key thing to know is to detect a change, to detect a stimulus, you would need to have some kind of receptor. In this case, we have a sensory receptor. Okay. So once it detects the temperature change, it is going to send the signal to the brain. And I'm sorry, I need to add more space here for myself. There we go. Okay, so all this stuff, we're gonna send a signal to the brain. Right, so there's gonna be a path here. Okay. That sends the signal all the way to the brain. Now, what is this path called? This is called the afferent pathway. Right? And it is made up of sensory neurons. Okay, what does it do? It carries the afferent pathway carries sensory information towards the central nervous system. Okay. So I'm going to put a dot here. Okay, so all these things that are happening here, that's part of your peripheral nervous system. That's part of your PNS. And so the signal, the sensory information, is going to carry along these sensory neurons, and then it's going to go to the central nervous system. Right? And in the central nervous system, you're going to have another kind of neurons. Right? It's going to be receiving the signal. And this one has like two tails. Not super important that you know that. I'll write something for you in a second. So this path here, right? It's a bundle of axon, right? What do we call a bundle of axons in the PNS, guys? Look it up. What do we call a bundle of axons in the PNS?
Nerve.
Nerve. That's right. And this nerve carries sensory information So we call this a sensory nerve. Okay, so all that information is going to be going towards your central nervous system. You touch the hot surface, the signal is going to go all the way up the sensory nerve, and then it's going to go to your spinal cord in this particular case. So the central nervous system is brain plus spinal cord, right? Brain plus spinal cord. So the afferent pathway is made up of sensory neurons. Now, inside the CNS, we're gonna have these special neurons with the two tails. We call them interneurons. Right, so what do? The interneurons, they are only found within the CNS. Only in the brain or the spinal cord, and their job is to process sensory information and determine the appropriate motor output. It's going to analyze the sensory information and decide what to do with it. Sometimes the appropriate thing is to do nothing about it. Okay. Well, in this case, obviously, you have to remove your hand, right? Okay, but like in other instances, maybe the best thing to do is like not to do. Like, you know, if you see a bear while you're hiking, like don't run, right? I think the advice is like to stand still. But if it runs towards you, then you run or something like that. Okay? But my point is like sometimes the best thing to do is not to react to it, right? Not to react to a certain stimulus. You don't have to necessarily respond to all the stimulus that's coming into the body, right? Okay. So story so far, are we together? Do you understand? You know, with the PNS and the CNS, right? So like Madison, you asked earlier, right? Like you said you didn't quite understand what it means by carrying signal towards the CNS. Like, is this a little bit better, or you have like a follow-up question?
I think I'm getting.
All right. So let's keep going for now, and then I'll tell you guys more. So now, what's the appropriate action here, right? Obviously, to remove your hand, right? So the interneuron is gonna send a signal back to the hand in this case. Okay, it's more like to the arm, right? But like you get it, like to remove your hand. So this here, this is called a motor nerve, right? And the name we give it is it's the opposite of afferent. It is the efferent pathway. Some people pronounce it efferent. Same thing. It's like tomato, different pronunciation. This is made up of motor neurons. And the motor neuron, they carries motor information from the CNS to effectors. I'll explain what that is in a second. An effector, not to be confused with the effector we learned in the immune system. This has absolutely nothing to do with it, okay? It's just the same thing. An effector is a muscle, gland, or an organ that carries out the motor instruction. So like in this case, the effector is the hand, right? Like to get out of the way from the flame. So I have several more things to tell you once you finish copying. I'm just gonna go to the bathroom quickly, okay? You guys keep copying what you need to. I'll be right back. Sorry about that. Okay, so tell me, how else would you feel like after you get burned accidentally? Okay, like think about last time this happened to you. Okay? So that you remove your hand, that's what this describes. You know, how else do you feel?
Pain.
Yep. So there's going to be pain, which also is part of this pathway, as you will see. But like maybe your heart rate goes up a little bit, right? Okay. Because you get scared, right? Okay. So
Yeah.
these motor nerve pathways, they're not necessarily just going to go to like one single target. It's possible for this to branch off, and then it's going to go, cause your heart rate, increase heart rate. Right? It's possible to have multiple outputs. And same thing, when it comes to input, we can have multiple sources of input. But I keep it simple with the hand in this case. Think about driving. When you're driving, the decisions that you make, right, is based on many things, many information that's coming in, right? Your eyes, right? You might hear sound, right? And then the pressure you're feeling from the accelerator or the brakes, all these information is going into your brain. You're making calculations subconsciously in most cases before you know whether to change the lane or not, right? Okay. Maybe you're going too fast, you see the cop, and you're like, oh no, right? Then the reflex is to step on the brake, and then your heart rate goes up. It's like, I don't want another ticket, right? So all these things are basically involving the afferent pathway, the interneurons, and the effector, as well as the efferent pathway. Okay, so again, back to your question in medicine. You were asking, I think, if I remember correctly, whether the signal originates from the CNS or not. Not all the time, right? So this description gives you an example of where it starts with a sensory receptor. Right? And that's true for a lot of things. Like, when your eyes see certain things, the eyes are serving as the sensory receptor. What you're seeing gets sent to the brain, and then, you react accordingly. Okay. But we don't function based on reflex alone. right now, like I'm talking, I'm teaching, right? I have to make up the words and then I'm moving the hand by my own will. At least I think I am, right? So those kind of signals comes from my central nervous system. Right? So it's possible for the signal to initiate from the CNS, and in this case, we bypass the sensory neuron. Okay, any kind of voluntary movement in the body, as we will see in lecture 6, starts from a special part in the brain that creates that signal, and that signal gets sent down the spinal cord and then to whatever muscle you're trying to move. So in that case, only motor neurons are involved and not the sensory. Any questions at all?
Yes, Professor. When the signal comes into the CNS, it goes to spine and then to brain, right? Because this is a little bit confusing because you just showed only one interneuron.
That's right. That's right. So that is a great question. We will look more into it in lecture 6. But short answer to that is it comes into the spinal cord, and then the spinal cord sends the signal back out to the hand so you can remove it quickly. But at the same time, the signal also split and goes up to the brain. But the signal to go out to your hand is faster than the signal is going up to the brain. That's why in most cases, we remove our hand and then we realize what happened. Right? It's almost always that by the time you realize, oh, that was hot, I got burned, that your hand is already off the surface, right? Do you understand? So signals can split and go to multiple pathways. I'm just showing you like a simple circuit here to help you understand. Another thing that I want to bring your attention to. In this note here, I said that dendrite receives signal from another cell, usually a neuron, right? And here it says that the axon terminal sends to another cell, sends signals to another cell, usually a neuron. So what is that exception? Do you guys know the game like Broken Telephone, right? Where you have a bunch of people and then you pass the message from one person to another person? Okay. So this is kind of like that. Within the afferent pathway and the efferent pathway, the signal actually basically get passed from one neuron to the next neuron to the next neuron to the next neuron, right? So within this chain, you're always receiving from a neuron and then passing it on to the next neuron. However, the chain has to have a beginning and an end, right? So what's the beginning? The beginning is the sensory receptor. That's the one that initially generated the signal. Right? So if you are the first neuron in the whole path here, the first neuron in this whole path, you will be receiving the signal not from another neuron, but directly from the source, directly from the sensory receptor. Okay? Yeah, so like a domino, right? So like the first domino is not being pushed over by another domino, but in fact, it's being pushed over by like the finger, right? Like there has to be some kind of trigger. So here, the first trigger is the sensory receptor. Right? So if you want, you can put it here, usually from a neuron, okay? But like the first neuron in the pathway would be receiving from a sensory receptor. I'm not sure how to add it here. I don't want to make it additional, make it even more messy. And what about the end, right? If you are a neuron that's within the chain, then yes, you'll be passing it on to another neuron. But if you are the last one, if you are the very last neuron in this path, what will you be passing the signal on to? Tell me the right term here, guys.
Organ?
Yeah, it could be an organ, right? It
Or
could
body
be
part, you can say.
it could be a muscle, could be a gland. So in general, we call that an effector, right? So a neuron usually passes a signal onto another neuron. Unless you are the last one, then you will pass it on to an effector. All right. We did so many things in the last hour. Any questions about any of them? Now would be a good time to ask.
So the first impact we are doing to removing our hand is coming from spinal cord, not the brain.
Okay, so just so that I'm understanding you correctly, you're asking what is responsible for triggering you to remove the hand? Is that what you're asking?
Yes.
Yeah,
Yes.
in this case, yes, the spinal cord.
I have a question.
Yeah, go ahead.
The exon can be messaged to immune system for help to like when I cut my finger and exon send some message to my immune system and help to
No. So this not at all. The immune system is something completely different. Okay. So like if you cut your finger, right? It's the damage to the tissue that triggers your immune system, right? It's not the nervous system in this case. Okay. Anyone else?
So the nervous system is control only motions and stuff. The immune system is just controlled by their immune, like all the system. It's not has an effect by the neurons.
No, that's right. Yeah.
Okay.
I have a question.
Mm-hmm.
So your example with like the heart rate going up if you get burned, would another like branch off could be like saying a certain word as you get burned?
That's right. That you know no, but actually that's interesting. Like if you curse, right? If you burn your hand, you're like beep beep beep, right? Those cursing are more like voluntary stuff in most cases, right? So that would be originating from the CNS, from the brain. Okay. Also if you burn your hand, right? You might blow onto it. You might run it under the cold water. All these things that comes afterwards. That's like based on your previous experience, and it's more like a choice rather than a reflex. Okay, remember, reflex requires no prior learning. Nobody taught you to remove the hand. Like your body just automatically knows how to do that. Does that make sense to you?
Okay, yeah, because like you saw someone else do that, so it's like, oh, I figured like I'll do that too when I
That's right.
start getting cut.
And those are again choices that you make, and it's not part of the reflex. Okay, reflex would be like you have no control over it. Like you burn your hand and then you automatically go turn on the faucet and put your hand down, and you couldn't stop that. Like, but that's not true, right? Like you deliberately chose to do that, and those kind of things originates from the brain, from the central nervous system. Okay, here is another example. Sorry, what's that? Go ahead.
So when those people just like go to like burn themselves on purpose, how do they like defy like the automatic response by leaving like their hand or whatever on the hot surface?
I mean, like we could too, right? Anybody could do that, but there's going to be a threshold, right? Like you can. So that's why like the candle might not be the best example. Like we all could deliberately hold our hand over the candle, okay? But there is going to be a point, the threshold where the pain is too great, and then it triggered the reflex, right? So we have voluntary control to suppress for a little bit of time, but eventually the reflex is going to kick in, right? Some people have higher tolerance to pain than others, and they might be able to suppress it for longer, but eventually, like the breathing that we talk about, right? Eventually your autonomic nervous system is going to kick in, and it's going to override your voluntary input, right? So here's another example. You're driving on the road, and I don't know, like in Canada, like a goose step onto the road, right? So you saw that. You see the goose coming out. So that's the eye being the sensory receptor here, and you're gonna send the signal to the brain. In this case, it's the brain that's doing the processing, not the spinal cord, because the eyes are closer to the brain than it is to the spinal cord, right? That's the only reason. And then it will send the signal back out to the calf muscle to contract and step on the brain. So in this case, the effector is the calf muscle. Understand? All right. You know what? Why don't we take a quick break, and then we'll take more notes when we return. Not too long though. Okay, let's just do a short one. Let's do 9:40, and then we'll return. Just a couple minutes for you to catch your breath. See you in a bit. Okay, so next we're gonna take a quick note on the division of the nervous system. Okay, division of the nervous system. Division of the nervous system. So we start with in the middle here, the nervous system. And we already know the nervous system is broken down into the central and the peripheral. But like, what does that actually mean, right? Like, so I keep on telling you the central nervous system includes the brain and the spinal cord, but like, physically, what does it mean? So this is the brain. This is the brain. Looking at it like from the back or something, okay? From the back. That's what the brain looks like. And then you have your brain stem here, and then you have your spinal cord. So this is brain. And then this is your spinal cord. And I don't know if you know, but the spinal cord becomes like a ponytail at the end, for real. So this here, that's the CNS, central nervous system. And then coming out of the brain, directly from the brain, I mean, not exactly like the way I'm drawing it here. Okay, but coming out of the brain, we have these nerves, right? Bundle of axons, right? These are called cranial nerves. And then coming out from the spinal cord, we have other nerves. These are called the spinal nerves. And these are part of the PNS because they are still part of the nervous system. They still carry electrical signals for you. Okay? But they're no longer within the brain. They're no longer within the spinal cord. They are part of the PNS. Okay. Let me show you a quick picture of that. So, this is the brain, and then the spinal cord is very thin. It's within your backbone, and we have 12 pairs of cranial nerves and 31 pairs of spinal nerves. It's easy to remember. It's like there are 12 months in a year and 31 days in half of the months, I guess. So coming out directly from the brain, that's where the cranial nerves are. And the 12 pairs, they do many things. Okay, including you have like 4 pairs dedicated to eye movement alone. You have some that will be dedicated to swallowing. You have some that are dedicated to hearing. We're not going to memorize all of them. Okay? And then we have the spinal nerves, 31 pairs. Okay? Some exiting at the chest area. Okay. Do you know how many pair of ribs you have, guys? How many pair of ribs?
12?
Yeah, that's right. So like the ribs which are at the front, they go to the back and connect to the backbone, right? And then where the backbone stacked together in between the ribs, that's where the spinal nerve is going to come out. We'll learn more about that next week. Here are the cranial nerves. Do not need to memorize this, okay? You don't need to know this. All you need to know is that there are 12 pairs. But I was telling you, right? Like you have a bunch of them dedicated to eye movements. Okay, 3 pairs, not 4. 3 pairs dedicated to eye movement. You have some dedicated to, the muscle of the face to allow you for facial expression, for hearing, so on and so forth. If you become like a nurse, I think you would have to learn the different kind of tests you would perform on a patient to identify whether or not they have a particular a defect in one of these cranial nerves. Okay? There are certain tests you can do to see if there's something wrong with the trigeminal nerve. Okay, so on and so forth. This, by the way, this is also the one that where the herpes virus hides in. The one that gives you like the cold sore, and then it comes out every now and then. Just a random fact. Okay, back to our note. So, what's going on with the PNS? Well, the PNS can be further divided. Although this division is not like an anatomical division, but more like a functional division. Okay, functional division. What that means is like we're not dividing it because the PNS physically have certain things here and physically have something over here. It's not that, right? They are kind of all over the body, but in terms of function that they carry out, it's different. So we have a branch of the PNS dedicated for autonomic stuff. We call that the autonomic nervous system, right? Or ANS for short. Yeah, ANS. So this is dedicated for like involuntary movement. It's dedicated to many automatic processes that happens in the body, like regulation of your heart rate, your breathing pattern. Okay. The other division is for voluntary stuff. Okay, voluntary stuff, like moving your muscle, talking, and this is the somatic nervous system, or the SNS. So just very briefly, this is for like voluntary, and this is more like involuntary. Right? But it's not so clear cut sometimes, right? Like in a reflex situation, the one that I described to you, the fact that you remove your hand after you got burned, that's an autonomic response, right? But does that mean I have no voluntary control over my hand? No, you can move it too, right? So like the same thing can be what's the word? Can be influenced, right? By both the ANS and the SNS. Okay, but like in different situation, in different scenarios, right? All right. Within the ANS, this autonomic nervous system, there are two other divisions that work against each other. And we have the sympathetic nervous system. I have the PowerPoint slides coming up, but very briefly, this is for the fight or flight response. Fight or flight response. And then we have the opposite, which is the parasympathetic. Nervous system. And this is more like, rest and relax state. Now I'll explain. Okay, so these two systems, they are opposing each other. They're antagonistic. Antagonistic. Like in English, like a story as a protagonist, antagonist, antagonistic, opposing each other. Okay. It's not like you will either have the sympathetic on or the parasympathetic on. You can have both sending the same signal to the same effector, but it's a balance. Like which one is dominating? That's going to determine the outcome, right? Any questions with the division so far?
Can you explain fight response one more time?
Which one, sorry?
Sympathetic.
The sympathetic and the parasympathetic. Yeah, I'll go through that in the PowerPoint slides. Yeah. All right. So the ANS, the autonomic nervous system, has two subdivisions, right? Sympathetic and parasympathetic. And as I say, they exert antagonistic opposing effect on the same organ. So you can see they both go to the lungs, and they both go to the heart, but one will speed up the heart rate while the other is going to slow down the heart rate. It really depends on what situation you're in. Sympathetic division, like I said, is for fight or flight. It's most active during emergency situation. You're walking in the park, and then this angry looking dog is running towards towards you. Then your body activates a fight or flight response. Do I fight this thing or do I run away from it? Either way, the response require a lot of energy. So the sympathetic system is going to increase your heart rate. It's going to dilate your bronchi so you can increase your breathing, dilate your pupil so you can see better, increase energy output. This is not the right time to think about digesting your food. So you will inhibit your digestive system. Okay. But the fight or flight response can get activated when you are like super excited. Okay, like on a roller coaster ride. Your heart rate also goes up. You feel a sudden surge of energy, right? So it's not just in a dangerous situation. And sometimes it's also possible to have the fight or flight response or the sympathetic division activated when you just feel stressed out, but you're not actually in any danger. I remember when I had my first job interview, I was so nervous. My mouth was dry and I couldn't talk. That's my sympathetic system being activated. Yeah, drying of the mouth, right? That's the suppression of the digestive system. Okay, like my heart rate is going up, feeling sweaty, feeling nervous. So that's sympathetic system. Okay, I sometimes have students who are so stressed out on the test that they feel sick. Right? One person vomit because of the stress. She told me she was not sick, but then the test made her feel sick. I feel kind of bad. I didn't know the test was like so challenging and stressing people out. But you probably can relate, right? in your life. In some situations, you just feel this anxiety. That's the sympathetic division. The opposite is the parasympathetic. This is the rest and relax, the rest and digest situation. Okay, you're just chilling. There's no immediate danger. Okay, you're relaxing at home. Heart rate goes down. Pupil gets smaller. Now is the time to promote digestion. Okay, sometimes this is called a housekeeping division. Right? Inside the body, it's time for cells to heal, to grow, and to divide, so on and so forth. Right? That's why kids who grow up in a stressful environment, like a war zone, for example, their system is always tilting towards this sympathetic division, and that leaves them very little time to grow, to heal, and that might stun their growth during critical times in a person's life to develop, right? So are we okay with this idea of sympathetic and parasympathetic?
Yeah.
Yeah. Anyone else have any questions? Okay, I have questions for you then. Which of the following is or are part of the PNS? Okay, very good. Basically, anything that's not the brain or spinal cord would be part of the PNS, right? Okay. Even well, actually, most of you got it right, so I don't have to explain. Just no brain, no spinal cord. You're good to go. All right, next one. The PNS is divided into. Let's do that one more time. What do you think?
Professor, are we talking about the system only, or there's parts involved only as well?
Are we talking about the system only or what? Sorry?
There's a parts as well, like the afferent and efferent nervous system. Those are the parts, and the other ones are system.
Right. Yeah. So like the afferent and the afferent is part of the PNS as well. Yes. Okay, I see what you mean. Hold on. Yeah, let's finish this question first, and then we'll talk about it. All right. So according to the notes, right? According to that, what do you call those things? The flow chart or whatever. The thing we just drew. Okay.
Yeah.
The PNS is divided into where is this? The ANS and the SNS, right? The ANS and SNS. Look here. Right? Literally. PNS is divided into ANS and SNS. Okay, that would be the most correct answer.
But also, Professor, when we draw the previous picture, afferent and efferent nervous system is also part of PNS.
Yep. So afferent and efferents are not system per se. They are pathways. What's the difference, right? Okay, let's think about this. Let's say we use touching the hot candle, right? This is actually a good question. Right? So like when you touch the hot candle, which of these here is responsible for picking up that signal? Well, if it's from a part of the body that you have voluntary control over, then it's going to be from the SNS. Don't draw this on your diagram. I don't want you to like, mess it up. But like touching hot surface. Right? That's gonna be picked up by the SNS. It's gonna go to the central nervous system, and then whatever motor response is gonna come back out here through the SNS so that you can remove your hand. Okay? So the blue path is the afferent, AFF, and then the red path here is the efferent, the EFF.
Yeah.
Now, what if the signal originates from something that you normally don't have voluntary control over? Let's say you ate too much, okay? And your stomach is hurting because it's like, too big, it's too full. I don't know if that's a good analogy. Like or like, if someone has like kidney stones or something, okay, and it's hurting. Now you don't have voluntary control over your stomach or your kidneys, right? So don't, but you can still feel pain from them. So that kind of pain is going to be picked up from the ANS, right? So it's going to send a signal to the CNS, and then it's not so much of a reflex in that case, but then it will send some signal out to the SNS here to, for example, like rub the stomach a little bit, or like you crouch over, or you take medication. So like the origin of these signals that detects the changes could originate from SNS or ANS, depending on whether you normally have voluntary or involuntary control over these body parts. Does that make sense to you? Or
Yeah.
yeah. Okay. And sometimes the whole thing could be ANS, right? Like your body detects higher carbon dioxide level, that's ANS. Goes to the central nervous system, the brainstem, and then it changes the breathing pattern automatically. That is still ANS, right? So it's not restricted to SNS or ANS necessarily. There could be some kind of crossover. I hope I'm not making it worse, but that's the explanation.
Yeah, that's good. Thank you, Professor.
All right. Anyone else? Like I haven't heard from like 70% of the class for the whole semester. Are you guys okay? Okay, like anything do you want to ask, follow up? Because we are about to take the very long note. Okay, thank you. This is a very long note, okay? And it's also a confusing one. You have to ask questions along the way if you are lost at any point. It's not going to go away. As in like, you're not going to be able to figure it out later on. Okay, just ask me on the spot if something doesn't make sense. Now I need you to have 2 pieces of paper for this note. One piece, we're gonna draw some stuff, we're gonna write some notes, and then the other piece, we're gonna draw a graph. Okay, we're gonna draw a graph on it. The graph's gonna be related to the stuff that we are going to be explaining, okay? So yeah, what is this note called? This note is let me see what I call it in the PowerPoint. Okay, action potential. Yeah. So this is action potential, which is a mechanism or a signal for impulse. Okay, we'll use a proper term for impulse to travel down an axon. Okay. Homer Yun was asking, how does the signal jump, right? Along the axon. What's triggering it? The thing is called action potential. We're going to learn about that. And it's a really confusing process. So let's start with some background information. Background information. Okay, background information number 1. The sodium potassium pump. If you were with me last semester, I briefly talked about the sodium potassium pump during transport, okay, membrane transport. If you don't remember anything about it, that's okay. So the sodium Na plus potassium pumps are found along the axon membrane. Remember, the axon is part of a cell, and therefore it also has a membrane, right? So along the cell membrane, you would have these sodium potassium pumps. Their job is to uses ATP to pump 3 sodium out of the cell in exchange for 2 potassium into the cell. So here is the neuron. We're just going to take a piece here and zoom in. I am not drawing the myelin to keep it simple. Okay. So big drawing here, guys, if you could. This is the cytoplasm, the inside. This is the outside. Okay.
Is it the axon we are drawing, Professor?
Yes, this is the axon. This is the inside of the axon, and this is the outside environment. Okay, look, I'm basically like zooming in here, right? Zoom in. This is also outside, huh? Here is gonna be our sodium potassium pump. There we go, a bunch of them. And we are going to be pumping sodium out. Okay, 3 sodium out. In exchange for 2 potassium. Let's just take a moment, draw that. So over time, what's going to happen is, I hope you will be able to see that, you will have more sodium on the outside, right? That's why you need color. Okay, if you're doing the dots in pencils, it would not make as much sense. Does that mean you have no sodium inside? No, you still have some, just not as much. And then for potassium, it is the other way around. Okay, potassium is the other way around. Okay, if all these dots are like confusing you, you can just write low sodium concentration. Remember, square bracket means concentration on the inside and high sodium on the outside. Okay, I'm gonna repeat it over here. You will see why later on that I have to write it on both sides, okay? As for potassium, oops. As for potassium, it is going to be high on the inside, but low on the outside. Okay, so, copy what you can and, ask. So I have a very important question for you to consider. Think about it. If the sodium potassium pump continues to do this, and it will, that is to continue to pump sodium out 3 at a time, but only bringing in 2 potassium every time. Which side of the membrane is going to be more positive? Is the outside more positive or is the inside more positive? Think about it. Now, don't tell me the answer yet. We just think
Outside?
about it. Okay. Let's do a vote. Let's do a vote. My question is, as this keeps on happening, is the outside more positive or is the inside more positive over time? Please let me know. Oh, did I not? Oh, I didn't launch the poll. I'm sorry. There you go. Which side is more positive?
I have a question, Professor.
Yeah.
Once the sodium goes out, it comes back in through those turbines.
No. You're confusing with the H plus stuff in the mitochondria. This is not that.
Okay.
Yeah. It's okay to not know here. Okay, yeah, I'll throw in a third option. Not sure. Okay, well, 9 people, 10 people. 70% of you said it's positive, more positive outside, and then some of you say it's more positive inside. Well, let's think about this, right? Okay, we are pumped, not we, but like the sodium potassium pump is moving 3 sodium out. Each of them is positive, right? So you're losing 3 positive, and you're only bringing in 2 positives. So each exchange means you are losing 1 positive to the outside, isn't it? You put 3 positive out, and you're only bringing in 2 positive. So each exchange means you are losing a positive. So over time, it is going to be more positive. Oh my God, that is too thick.
Very positive.
Well, I wouldn't say very positive necessarily, but it's gonna be positive, okay? It's gonna be positive on the outside. Positive on the outside. And then it's gonna be negative on the inside. Okay. And I, again, cannot stress the importance of using colors here. So as the sodium potassium pump operates, the inside of the membrane is negative relative to the outside, creating a membrane potential. I will explain what that means in a second, okay? Just copy it down first. Creating a membrane potential. Membrane potential refers to the charge difference across the cell membrane. It is measured in millivolts or MV. Okay. The measurement is always the inside relative to the outside. Okay. What that means is, instead of saying it is positive, we're gonna say it's negative. You see here, right? Like when in my drawing, I have the negatives on the inside and the positive on the outside, right? So we're always describing the inside of the cell relative to the outside. Okay. For those of you who don't really quite understand the concept of like volts, you can look at your battery, right? Okay, like the standard like AA battery. If you take a look at it, it will tell you it's like 1.5 volts. Right? You have the bigger ones, right? These ones, the 9-volt battery. Right? So V volts is basically a measurement of the charge difference between the positive and the negative terminal. Okay, so those charges are separated in a battery but only like when you hook up the positive and the negative, then you have a flow of a current, right? So in this case, the positive and the negatives are separated by the membrane. Okay. And there are no paths for these ions to flow. It's same as a battery that has no wire connecting the two terminals. The current is not going to flow. Now I want you to think about this. This is important. Okay. When given the chance, which way will the sodium flow? Which way will the red dots move?
Inside?
Inside, that's right. Because things have a tendency to go from high concentration to low concentration, right? So the sodium would want to come into the cell. But it cannot do that because it's charged, and then the membrane does not allow charged particle to go through, right? Same thing for the potassium. It's very high on the inside. It wants to leave the cell. It wants to go out from high to low, but it cannot because the membrane prevents it from happening. That's why we're creating this membrane potential. Right? If there are holes along the membrane, then all these things will just go back to equilibrium, right? Go back to balance. But the fact that they're separated, and then we have the sodium potassium pump keeps on pumping them against their concentration gradient, we create this uneven distribution of ion. And this uneven distribution creates the membrane potential. Does anybody have any question about what membrane potential means? Okay. When the neuron is at rest, at rest means that is not conducting a signal. The resting potential is negative 70 millivolts. This number is going to be important. Okay, I'm going to throw you a few important numbers throughout the class, throughout the notes. So the membrane potential, again, is just the difference between the inside and the outside, right? That number can change depending on what the neuron is doing. When it's not doing anything, that number is minus 70. We call it the resting potential. Okay? So think of membrane potential as just like a broad term. But if you measure it at different times, it's going to give you a different number. Membrane potential is like the word temperature. But then, you can have like room temperature, right? Ambient temperature, outside temperature. When you measure at different times, different locations, it's going to give you different numbers, right? So the membrane potential, when you measure it, when a neuron is not doing anything, the number is minus 70 millivolts.
Professor, I have a question.
Mm-hmm.
On the resting potential, the pumps are doing their work. So it will not be keep on increasing because of the charge will keep on moving from one side to the other.
Yep, that is a good question. Short answer is no, because there are other counteracting mechanisms to balance this, but that's beyond the scope of our discussion. Okay, there's actually like a math equation that shows you, other counteracting forces, and then they all equilibrate with each other, and it becomes minus 70 in the end. Okay.
Thank you.
It's like there is almost like a overflow valve that if the voltage goes too high or too low, the ion is going to be able to rebalance themselves. It keeps it at minus 70.
That makes sense now.
Okay, that is background information number 1. Everything good? Yeah. Move on to background information. That was background information number 1. Okay. Now we're gonna do background information number 2. Number 2. Okay, which is called voltage sensitive or sometimes called voltage gated ion channels. Okay. That requires you to kind of remember something we learned in the last semester, right? Let me see if I think I have the note recently from my other class. Is it lecture 4? Yeah, probably lecture 4, right? Okay. Lecture 4. Remember this stuff? We did this last semester, right? And under facilitated diffusion, there's something called like a channel protein, right? Channel protein allows ions to pass through. Well, now is the same situation, but these ion channels, they are voltage sensitive, okay? Or they're voltage gated, which means whether they are open or closed is going to be dependent on the voltage of the membrane. Okay, so there are two types that we need to worry about in this situation. Two types in this situation. Okay, we have the sodium. I'll keep on using red for sodium. Sodium channels that are voltage sensitive. Okay, I'm not going to write the whole thing. Voltage sensitive sodium channels. So there are two configurations. This is the cell membrane, and this is the voltage sensitive sodium channels. Okay, think of them as like a door. So this is close. And obviously, when it's closed, nothing goes through. And then this would be like open. Okay. So when it's open, the sodium can move through them. Okay. When it's closed, nothing passes through. The opening / closing of the channel depends on the membrane potential. Okay, this is open. At -70, all sodium channels are closed. All sodium and potassium channels are closed. Right? So when do they open? For sodium channel, it will opens at -60. And then it will closes at, I'm rounding the numbers so that it's easier for you to remember, okay? At around 30 millivolts. Okay. So each of these channels has an opening voltage and a closing voltage. Think of them as like the door to a store. Right? They will open at 6 a.m., but they will close at 3 p.m. Right? Once they're open, the sodium can pass through. I lost my blue color. Where is it?
Professor, what do you mean by the minus and plus here?
Okay, so the minus is, so right now when the neuron is not doing anything is at minus 70, right? And then I will tell you something is going to happen if the membrane becomes minus 60, then the sodium channels will open. If it stays at minus 70, nothing will happen, nothing opens. Okay,
So if it is keep on going back from 0, then it goes to plus.
that's right. Eventually, after it goes to 0, it's going to go like, 10, 20, 30. And if it's at 30, then the sodium channel will close.
Oh,
Okay, yeah, it will make sense when we do the graph. Okay, so potassium channels. Okay, so we have two states. Okay, it closes at minus 90. I know it's really confusing. Because like some of you might be wondering, wait, if it closes at minus 90, then why is it close at minus 70, right? I'll explain. Okay, just give me a second here. And then this one is going to be opens at positive 30. Okay, and when it opens, then potassium can move through, right? Of course, the potassium can only move through the potassium gate, and the sodium can only move through the sodium gate. Okay. So you cannot close the door unless you open it first, right? So when you are sitting at minus 70, it's already closed. Okay? You cannot open them until 30 millivolts. Once you open it at 30 millivolts, then you can close them again at negative 90. Okay? If you don't fully understand about this, how the numbers work, it's okay. When we draw the graph, it will make sense. Okay? But for now, that's all I need you to know. Just be aware that they are closed when we are resting, but they could open at a certain number, but they don't open forever. They will close again when you reach certain numbers again. Okay. There is another thing here that you should know. Once a channel open and close, opens and closes, it enters a refractory period. During which it will stay close and become unresponsive until it resets. Okay. So it opens and closes. Opens at, let's say we talk about the potassium one, right? Opens at positive 30. Potassium goes through it. And then you close it at minus 90. Now it is in a refractory period. Even if the membrane potential reaches 30 again, it's not gonna open yet. It's gonna reset itself first. Okay? Let me say that one more time, because I think some of you might have missed that. Sodium channels, right? Okay, close at resting. Let's say it opens at minus 60. Okay, and then the sodium's gonna move. And then when it's 30, for some reason, it will close. And then it will stay closed. It will enter a refractory period. During this time, even if the membrane voltage goes to minus 60 again, is not gonna open because it's resetting itself, right? Only after it resets itself, then it's ready to respond again. Okay. So that is that. Okay, that's all the background information. Let's just take a quick break. Well, I think we probably have to go beyond. We'll see. Okay, because I don't want to not finish the note. You know, probably have to stay a little bit longer today. Okay. But let's take a quick break first. Okay, for you to just go through the materials, let it sink in, and we'll resume after. Now, we're gonna take a look at what happens when the signal needs to go down the axon. We have all the background information that we need. And this part is a little bit tricky to teach because, like, you know, I need to do multiple diagrams. And you know, I don't actually have enough board space here on the screen than I would in the classroom. So I'm gonna have to move back and forth a couple of times. You know, if you're confused, just tell me to slow down, right? But like, I need you to have the piece of paper next to you for the graph. And you know what? Let me open a new page here for the graph specifically. Okay. So this is the graph that we're gonna have right here. I need you to big one, okay? Big graph. This is your paper. Okay, big graph. Right? So right there. All right, so like on the y-axis here, on the y-axis we have the membrane potential, which is measured in what units again? Trying to keep you guys engaged here. What's the unit?
Millivolt?
Millivolts, yeah, MV, yeah, millivolts. And this is going to be time. Right? And it's really fast. It's measured in milliseconds. You know it's really fast. When you touch a hot surface, your hand is like removed in seconds. The time it takes to pick up the signal and to respond to it happens within a second. That means the signal is going through your neurons at very, very fast speed. It's about 100 meters per second. Okay. All right. On the y-axis, where do we start? And we're gonna start. This is not to scale, but it's okay. It's not important that it's not. We're gonna start at minus 70. Minus 70 volt. This is the resting potential. Resting potential. What is happening to the neuron at this point? Absolutely nothing. Okay. No signal is being transmitted. It's just sitting there. It's okay for a neuron to not be transmitting signal. Okay. Like, I don't feel any pain from my hand right now. So, you know, there's no pain signal going through the neurons, which is a good thing. Right? Hopefully that's the same case, right? So at this point, we are gonna have two things here. Why is the color not changing? I think I'm writing too many notes. That's why the program's not working. Hold on. Okay. So at the resting potential, what can you tell me about the sodium level? And we are always talking about it from the perspective of the inside. Okay, unless I tell you otherwise. What can you tell me about the sodium on the inside? Is it high? Is it low? What's going on?
It's low inside and high outside.
Right, but again, I'm only talking about the inside, okay? Only talking about inside.
Oh, so it's low inside.
Yeah, so low inside, yeah. And then for potassium,
It's high.
it's high. Why? Because of the potassium pump, right? Sodium potassium pump. The sodium potassium pump is pumping all the sodium out. We can keep them out and bringing in the potassium. Okay. So that's good. Now, something is gonna happen. I cannot tell you what that something is yet. It wouldn't make sense if I tell you now, okay? But something's gonna happen, and I'm just gonna call it like a lightning bolt. Okay, something happens. A lightning bolt here. Okay. We can think of it as like the arrival of a stimulus. Okay, the stimulus arrives. I'll tell you more about it later. Okay, stimulus arrives, and it's gonna push the membrane potential higher. So minus 70 is gonna become like minus 68, minus 65. Nothing's happening yet. Minus 62, minus 61, minus 60. Okay. When it hits minus 60, okay, and again, I will tell you later why it's keep on going positive and positive, okay? When it hits minus 60, this here, we call it the threshold potential. And once you cross this minus 60, it's a point of no return. The signal is gonna go. There is no stopping the signal now. Okay? Below minus 60, it's okay. You can still suppress the signal. Okay, but beyond which, it's gonna go. We call this the all or nothing event. You either do it or you're not gonna do it. Once you pass negative 60, there's no turning back. Signal is gonna go. Okay, so what's happening at minus 60? Let's go back to our note. At minus 60, if you recall, that is the opening voltage for sodium. That's right. That's the opening voltage for sodium. Up to this point, they all had been closed, right? But now we are at minus 60, and I'll tell you how we get to minus 60 later on. Once we get to minus 60, they are going to open. And so let's draw what happens. Okay. I need you to draw another one, okay? Another one. Don't use the existing one. It's like it's going to be too crowded. Now, the stuff that's going to be happening from now on, at this point forward, it's not, I mean, the sodium potassium pump are still there. They're still doing their thing, but they are not going to be contributing much to what's happening. Okay, so I'm going to not draw them to keep it like cleaner, the diagram. Okay. But a signal arrives, and what's going to happen is, as I told you, the sodium channels are going to open, right? Sodium channels are going to open. Okay. The sodium channels were there all along. We just didn't draw it at the other diagram, right? Because like it was too much otherwise. Now, the axon is really long. Okay? So the signal is only at the beginning, right? Like closest to the cell body. So only the sodium channels here in this section is going to open. The rest of them are still going to be closed. You have to understand this.
Like a valve?
Yeah, like a valve, okay? But like, you know, we only opening this stuff, but everything else is closed. Everything is closed. Okay. What about the potassium channels? Well, did we reach their opening hours yet? Opening voltage? Nope. So they're going to be closed. Okay. So tell me, what's happening when the sodium channel opens? Which way will the sodium move? Into the cell or out of the cell?
Into the cell.
Into the cell. Let's take a look here.
Professor, I have a question.
Sure.
The membrane has a potential, membrane potential.
Mm-hmm.
If it will change, it will not change throughout the membrane.
Come again? I don't quite understand.
The potential, the membrane potential is minus 70 down, move down to 60 MV, minus 60 MV.
Move up to
Now
minus
when-
60, yeah. Mhm.
Yeah, so it will not change across the membrane, or it will change only a part of membrane.
Oh, you mean it will not change across the whole axon, right? Is that what?
Yeah.
Yeah, that's right. It's gonna be happening section by section, as you will see. Okay, now for the rest of the class, right? Listen, right? Look at this picture. Do you see a lot of red dots outside? Right? All these red dots are sodium, right? And then we don't have a lot of red dots inside. As I told you, the sodium wants to come in, right? How come they cannot come in? Don't do this to your diagram unless you really want to. They cannot come in because the doors are closed, right? The sodium channels, the voltage sensitive sodium channels are closed. It's not open yet because what number are we sitting at right now? What is the number for the membrane?
70.
It's minus 70, right? At minus 70, none of the doors are open. Okay? These red dots, they are like people on Black Friday lining up, waiting for the doors to open. They want to go into the store, but they can't because the door's not open yet. It's not the right time. It's not the right voltage. Okay? So when it is minus 60, then the sodium, all these sodiums are going to come in. All the sodium's going to come in. Sodium, 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 sodium. Right? They are going to come in. They're going to come in like that. Okay? What is that going to do to the sodium level? What is that going to do to the sodium level? That's right, it is going to go up. Right, so now this is going to be high sodium, and the outside is going to be low sodium. Okay, perhaps I didn't draw this too well. Okay, I should take away some of these things, and I should move them inside. Does anybody have any question about why the sodium is so high in the inside now? Anything at all?
I have a question. One thing,
Mm.
not related sodium high.
Okay, yeah,
but
yeah.
we have a two state from axon. When is axon in the middle line and the end of the line. When in the midline before the axon, what happened for the gate canal, and in front of axon, what happened for the gate canal gate? It's open before and closed after. The move when the move axon cross to the end, behind the axon, the canal open, and in front the canals closed.
Okay, so over here, the sodium channels open because of the lightning bolt. It causes it to become minus 60, and then it's open. Okay. But further down the axon, over here, further down the axon, it is still minus 70 over here. The lightning bolt hasn't reached this part yet. And so the sodium channels are still closed. And at this part, it is still same as the beginning. Okay? So further down the axon, for the rest of the axon, all the way until the axon terminal, this is still going to be low sodium and high sodium on the outside. Okay, whereas over here in this current section, it has been switched, right? Because of all the sodium coming in. Now, I really need you guys to think about this. Okay, and you don't have to answer right away. Okay, just like think about it, give some time to people to think about it. What should we write for the potassium here? Is it high or is it low? Let's think that through.
Right.
Okay. Let's do a poll.
Low.
We have some high, we have some low. And when you think about, look at the original picture, think about what's happening. Do you think the potassium channel is A, high on the inside, or is it low? And it's okay to be not sure. I am asking you, what should I put here for the inside?
Professor, I have a question.
Yeah?
How come the sodium goes against the gradient without any energy involved?
It didn't go against the gradient. They came in, they went down the concentration gradient, didn't they?
But it should go balanced, like
It should what, sorry?
it should go balanced, in and out should go balanced, and it should stop moving.
Oh, I see what you mean. I see what you mean. Okay. So like it's higher than before now. Okay. It's higher than before. Think of it like that. Okay. You were saying it should go until it's the same inside and outside, right?
Yeah.
Okay. But no, like they're coming in. They're higher than before now. That's what you should think about. Okay. I have more people who think it is low than it is high. Okay. All right, let's go back here. Let's go back here. Look at the blue dots, right? Do we have more? We have more blue dots on the inside and outside, right? Do you understand? More blue dots on the inside and outside, right? They are like, go back to the Black Friday analogy. All these red dots, all these customers want to come in. And the blue dots are like the Walmart employees. They really want to leave, okay? They don't want these people to come in. They want to leave, but they can't leave. Why can't the potassium leave? Tell me. How come the potassium can't leave?
Because their point is not there to open the doors.
That's why the doors are closed. The blue doors for potassium are closed. They haven't reached that opening signal yet, right? So you got all the sodium pouring in, but the potassium has nowhere to go. They want to leave, they can't, because the doors are not open yet. The channels are not open yet. They don't open until what number? For the blue channels, they don't open until positive 30. Okay? Which causes the jump? What jump? Oh, like from node to node? Not quite. Okay, just stay with me. We'll get there eventually. Okay, so this is still low, high. Sorry, this is still high potassium. Okay, on the inside, and then low potassium outside. Relatively, right? Is that okay? All right. Let's go back to our lovely graph here. So what's going to happen? Well, at the threshold potential right here at minus 60, what happens? The sodium channels open, right? And with the opening of the sodium channels, all the sodiums, not all, but like a lot of sodiums are going to be coming in. Going to keep on coming in, right? And what is that going to do to our membrane potential? We were at negative 60, but with all these extra positive coming in, it's going to keep on going up, right? Okay. It's going to dilute the negative, right? It's going to make it more positive. And it's going to keep on happening until 30 millivolts. Okay, that's the closing voltage for the sodium, isn't it? Okay. Once you reach the positive 30, then the red door shuts. No more sodium can come in. Okay? That's why it keeps, it's not going to keep on going. It's just going to stop there. So this phase is called depolarization. Depolarization. right? And it's caused by an influx of sodium. A lot of sodium coming into the cell, causing the membrane to become positive. And then at the peak here, three things are gonna happen. Sodium channels close, preventing more Na plus from coming in. Okay. And then the other two things we'll talk about later. I don't think I can finish the note. Even if we stay until 11:20, I don't think we can finish the note. So I think this is probably a good place to stop. And we will resume on Wednesday. Okay. But before we go, any last minute question?
I have a question.
Yeah.
When the inside high calcium and high potassium, it's take a time or immediately open valve and close valve.
The sodium
For,
channels, right?
oh,
Yeah. Yeah. The sodium channels is
I
the one.
see in the x axis times you
Oh, in the
you
x-axis. Okay.
yeah x axis your draw you see
Time.
yeah a little take a time between the
Oh, like how long is it gonna take here?
minus 70 MV and 30 positive
Yeah. You're
MV.
asking me how long is this gonna take, right?
Yeah.
Yeah.
No,
This
no.
is
This when the going up and maximum.
2:30. Yeah.
Yeah.
Oh, you're asking me how long how much time
Yeah.
that's gonna be, right?
Yeah.
Like couple milliseconds. Okay.
Yeah.
Yeah. I don't have the exact number with me, but like couple milliseconds. Yeah.
But it's happened.
It happened. Yeah. Really quick.
Yeah. Okay. Thank you.
Okay, so I really encourage you to please go through today's lecture, right? Review it before Wednesday, right? You have to. Otherwise, when we build upon this stuff, and you're gonna be so lost. Okay, maybe redraw some of the pictures. Okay, watch the recording again. Go through some of the questions in the study guide, and we'll wrap this up on Wednesday. All right. Thank you so much for your time and attention. See you next time. Bye.